Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Welcome back to Neuroverse. Today's episode is on mice playing poker, probing the neural circuit mechanisms of economic decision-making. And to help us with this episode, we are joined by Dr. Ann Duan, who is a PI at the Sainsbury Wellcome Centre. She originally obtained her PhD in neuroscience at Princeton University, where she studied prefrontal and collicular contributions to executive functions with Carlos Brody, and then became a Simmons collaborator on the Global Brain postdoctoral fellow in Ning Long Shu's lab at the Institute of Neuroscience in Shanghai. She used circuit-level tools to investigate cortico-subcortical cooperation during decision-making, formation, and maintenance. Now, Anne is researching economic decision-making in mice, as well as social decision-making. Welcome to the podcast, Anne.
Thanks, guys, for the invitation. It's my pleasure to be here.
It's wonderful to have you on. I'm so excited to delve into today's topic. But first, I would like to ask what initially inspired you to research neuroscience and where your passion for circuit-wide computations came from.
Yeah, I guess thinking back, really my initial interest was maybe human psychology. I was very interested in people. I was very interested, even as, let's say, a teenager, very interested in thinking about what people are thinking, which then drawn to psychology. So I grew up in Shanghai, China, and then for undergrad, I went to the US thinking that I was gonna study psychology and maybe become a psychologist. And my mentor at the time, so I told her I was gonna be psychology and pre-med major. And she was like, "Oh, have you heard about neuroscience?" To be honest, at the time, I haven't heard of neuroscience. And then I took an intro to neuroscience course with her. She was an amazing mentor and lecturer. So I guess we all have one of these people in our life that somehow inspired us to go down a path that shaped our life significantly. And she was that person, Judy Grizel, my mentor from my undergrad. So basically, I was hooked. I took the class. I was like, oh, this is exactly what I want to study, what I want to understand. I like the idea of thinking the question from a psychology perspective, but be able to have the tools to really look internally into the brain to probe the neuromechanism underlying the phenomenon that we see on the behavioral level. That was really my undergrad sort of initial entry into this path. And I have to say, pretty much since then, it's just been, I don't really have a super interesting trajectory, to be honest. Since then, it's just been kind of very straightforward. Towards the end of my undergrad, I know that I wanted to do a PhD. So I applied to many different places, and in the end, decided to go to Princeton. Over there, I would say most of my previous research experience has been on behavioral neuroscience, a lot of behavioral study, also with some studies in drug addiction and so on. Some experience with thinking about cognition, but not a lot. I think my rotations during my PhD was the first time I really think about a system neuroscience approach to study cognition. And, you know, a lot of sort of my students or other early career scientists come to me and be like, how do I know what I want to do? I feel like for me, once I encountered the approach, the level, and the questions that really interest me, then it was super clear. So then I did my PhD in Carlos's lab. Yeah, and then I did a postdoc, which was also very different experience from my PhD because my PhD was I was in a very well-established lab, and I was a PhD student, so I was a junior. And during my postdoc, I was in a great lab in the Institute of Neuroscience, Ning Long Shu's lab. And that was a young lab that mostly are students when I was a postdoc. So that was a really interesting kind of different experience. I learned a lot. All of my mouse experience were from my postdoc. And then I think, I talk to other colleagues in terms of what do we think about as an someone towards the academic goal of becoming a group leader in terms of like, oh, do you think you can make it or things like that, right? And I think it's completely healthy to be like, at least for me, in my head, it was always that was my goal. But also I was aware of the reality, which is, I have the passion and I think I have the skill sets, but there's so much noise and luck going to the process that I never assumed that I would be able to do so, right? And I think that definitely added some stress to my sort of postdoc period because I know that was the next step. But I think I was okay to be things. I really hope that things will work out. But if things doesn't work out, then I'll figure something out. But at this moment, this is the most fun that I'm having, and I'm really sort of fortunate to be having it as a job, right? So yeah, so internationally, that was also quite interesting, right? I grew up in China and then went to the US for undergrad and my PhD. That was 10 years of my time over there. Academically, obviously, there was a lot of growth and evolution, but also on a personal level and my perspective towards different culture in the world. And that was interesting sort of to go back to China as an adult. Also, my husband, Jeffrey Ehrlich, who's also a group leader at SWC. So he's from Canada. We met in the US and him, oh, actually, he went to China first. He started his first group at NYU Shanghai. And then I started my postdoc at Institute of Neuroscience in the Chinese Academy of Sciences. That was a very interesting experiences in both our lives where I was home as an adult and almost have an international identity, like a global citizen in a way. And my husband was at my home, a foreign culture to him, trying to start his group. So that was a really multifaceted and interesting period of our life. Very exciting and a lot of new things and learning about each other and so on. So then towards the end of my postdoc, I was pregnant with our son. So it was definitely a little bit on the earlier side in terms of readiness to apply for a group leader position. Your question was, how did you get interested in neuroscience? And I gave you my entire life history. But maybe after this bit, you guys can hit me with other questions as well that I can answer more specifically. So then I really had to think about if I have the opportunity to start my own lab, what would be the thing that really get me to wake up in the morning and get excited about doing, right? Like I will be given this opportunity to pursue something that really interests me, but is maybe generally interested to other people as well. It's super exciting, but also the uncertainty of it is a little bit nerve-wracking, as you can imagine. So I was listening to a bunch of different podcasts at the time and think about what really kind of similar to your question, thinking back to my early years to really think about, okay, I have this neuroscience experience to prepare me as a scientist, but what is the origin question that got me into this, right? Because maybe that is the right starting point. That's when I really sort of like decided to have a turn of my trajectory instead of following exactly where my postdoc was leading, which was what a lot of people do, decided to take a turn, right? So I studied decision making during my PhD and my postdoc. But then I was like, okay, let's think about what kind of real life decisions that we make every day, but because of sort of a reductionist approach in order to control what we study and actually learn something about things, we've been reducing the complexity in the laboratory, right? But now as technology develop, both in terms of tracking and analyzing behavior, but also in terms of the number of neurons, the type of neurons, the different turning on off neuron technology that we can use now, maybe there's a new opportunity for us to ask question about more complex type of decisions. So that's when I start thinking about decision preferences, right? When faced with the same sensory stimuli, how do different people not only make different decisions based on that, but also even interpret different conclusion from the exact same, dare I say, facts, right? And we see that all the time. We see that during election, how we interpret the exact same facts in the opposite fashion. So decision preferences in the sense that, we express the association between the input and output in a different way. And there's not necessarily correct or incorrect decision, but more about how that reveals who we are, our history, our community, our belief system, our priors, and our future goals. So then the rest of the kind of scientific question that we are now pursuing kind of started from there. And I have to say, that was kind of my dream, right? If I get the uh, position to do this, I would love to pursue these. But like I said, it was a little bit on the earlier side in terms of applying for a job because my postdoc paper was on bioarchive but not published. And but since I was pregnant, so I was doing experiments. So I was like, well, let's apply to a selected number of places where the next year I was, if I didn't get anything I like, the next year I was gonna apply to many more places and more broadly across the globe. But that year I only applied for a few places that I really, really wanted to go. And Sainsbury Wellcome Centre was one of them. And all the stars aligned. Not only did we get this opportunity, and we have the double opportunity where me and Jeff can both work at Sainsbury Wellcome Centre to pursue question we're interested in. So that is the origin story.
Amazing. I love it. So many twists and turns, but I feel like it all does very neatly align. And I imagine that making the decision of moving back to China and then moving to the UK and everything, and
Mm,
the all of the variables that you have to consider being
yeah.
pregnant, your partner, I can see how it inspired you to pursue this question and not answer it in such a reductive way, because you're right, so many studies are done where it's very binary and quite artificial, not very naturalistic, even for mice, let alone for humans, where we are always bombarded with loads of stimuli. So it's cool how you were able to take inspiration from your life experience and really pursue what you were interested in and got into the amazing SWC, and it's such a nice environment to work in, and the incredible collaboration that you get with the computational side of things as well is so beneficial.
Yeah, yeah, it
Yeah.
was a really beautiful story that I love that moment where you were describing how you were forced to make a decision to choose what you wanted to pursue and then how you were contemplating decision making at the same time. Um,
Yeah, it's very meta.
yeah. And I always love hearing these stories, like these origin stories of how scientists come to be, because it is always a combination, as you said, of luck and coincidences and driven by passion at the same time, and things just need to align. And so, yeah, thank you for sharing that.
Yeah, can I just like you guys are saying some really good points, just to kind of make a comment on that. Well, first of all, the sort of reductionist approach, there's a reason why we've been doing it for so long, right? Which is that's at some point the only way we could thoroughly understand something. And I have to say, in my lab, I have sort of the whole range from extremely artificial, non-ethological, but super well-controlled study that we can really nicely interpret the results, all the way to maybe not the most ethological, but much more ethological paradigms where it's much more natural to the animals. But then we face challenges in terms of segmenting, understanding behavior and neuroactivity, right? So I think I'm not definitely not saying that there's like one approach that's better than the other, but I think in recent years we feel more equipped to move towards one direction while still maintaining sort of paradigms on the other extreme. And I think going back to one of the other points is that, you know, this is a very sort of meta process. And I have to say, my decisions of what to pursue my own lab, it was pretty big risk. It was a pretty big risk, not only because some of the questions we're pursuing are brand new questions. For example, how do mice make value-based decision making in multi-agent environment, right? When the actions of others are going to influence the outcome of your own actions. We don't really think about mice that way, but exists in the neuroscience community. But of course, fundamentally, these animals have to survive and adapt in very complex environments. And they of course have the ability to do so. So like when you're talking to maybe an ethologist, they'll be like, yeah, mice have the capability of doing this complex behavior. But of course, recently we started thinking about mice like that. So how to design a behavior paradigm that we can actually ask this question, it took us a while to really come to that, right? And the other risk will be, I've never done this before. Would people give me a job to do this? And would funding agency fund me to do this, right? Because when they look at this, they will be like, not only do you not have a track record of doing this type of research projects, very few people in the field have a track record of doing so. So that was kind of a big risk, but extremely luckily that hasn't really been a problem. And I think it's maybe fair to say that so far it's been going really well, both in terms of sort of giving this opportunity to start my lab at SWC. I think it really reflects the vision by the community to give me this opportunity to be like, we should be the people to support others to pursue really bold questions, right? And even in terms of the funding perspective, we've been also extremely fortunate in terms of getting funding, which is always a surprise because I almost feel like all the reviewers have to take a leap of faith. They're like, okay, you can do decision making in general, and this is a very convincing proposal. Maybe you can do this as well, even though you haven't done this before. So it was a risk that was so far, completely worth it. But it was nevertheless a risk.
And I would love to hear more now about the specific paradigms that you have managed to come up with to study economic decision making in these mice.
Yeah, so thank you so much for asking. So the general paradigm I wanted to, the picture I want to paint as a background will be like, not so much think poker was just like a silly thing, right? But if you think about like a hungry mouse in the wild trying to forage for food, a lot of the times there's multiple options, right? And which option you should pick, let's say, fallen nuts versus cricket, these type of options. One is maybe safer, the other one might be riskier, but if you catch the cricket, a higher reward, higher nutrition and so on, right? And the preference of these, whether you should gamble or play safe, it really, first of all, differs across individuals of animals. Second of all, is modulated by internal states, how hungry you are, external variables, is it winter, is it nighttime, right? So like these preferences are quite flexible. And another source of what we call risk from these type of decisions can be attributed to others, competing others in the environment, right? Like maybe cricket, but you see a dominant mouse also trying to go for cricket, and that potentially is not the correct option to go for at that moment. So then we have the multi-agent aspect of it. And finally, there's the aspect of what we call stochasticity in our behavior. What that means is that you want to maintain a level of unpredictability in your own behavior. So that's not always so easy for others to predict what you're going to do, because once they can do that, then maybe the answer of what they should do is clear, right? For example, prey capture, you can imagine the prey had to have stochastic behavior. So that's not so easy for the predator to predict what the next move should be. Negotiations and, stock market and how we interact with each other in general, and football, that's maybe the best sort of metaphor for us. So that's another direction, which is how do we behave with, some may say noise in our decision-making process, but other may say really advantageous noise so that we remain unpredictable in this multi-agent environment. The three main directions I just mentioned are kind of the three research programs in my lab. The first one corresponds to understanding neurocircuit mechanism underlying decision under risk. I'll elaborate in a second. The middle one will be trying to study how mice learn to become stochastic when playing against a computer opponent that's trying to take advantage of the predictability in the mouse behavior. And then finally, we have two freely moving mice. The first two paradigms I mentioned are HEFIX, two freely moving mice competing with each other in a very dynamic foraging environment that's computer controlled, so we can have trial structure, different stimuli that we can present. And the rule is very simple, it's a competition. However, on each choice, there's always two options, better or worse. And when you're by yourself, you probably always go for the better option. But when you're competing with another freely moving agent in this dynamic environment, on each trial, the optimal choice actually could differ, because let's say you're close to the worst option, the other one is close to the better option. However, the rule of the game is that the faster mouse is going to get the reward and slower mouse will get nothing. Now, sometimes getting a lower reward is better than getting nothing, and which also means your competitor might get nothing if they're slower. So you can see the complexity in this kind of dynamics, which is really natural behavior variance that mice display on session one, right? Like we don't have to teach them to compete. They really pick that up instantaneously as long as they understand the rule of the environment. Yeah, so these are sort of three different paradigms in my lab.
It's so amazing how we need this stochastic behavior and how a level of uncertainty is necessary for survival. And how I guess humans are constantly trying to diminish the uncertainty of our environment in order to be able to make decisions, but perhaps we should kind of go along with the uncertainty of our environment, and maybe that's what mice are doing better than we are in terms of managing their anxiety or managing their decision making. But yeah.
Yeah, that's a really interesting point. I think we do a balanced thing, right? Which we trying to learn as much structure about the environment as we can. And based on these structures, we can behave adaptively, right? On the other hand, we often behave stochastically. Maybe we just don't think about it when we do. Take football, for example, right? There's so much stochasticity that's important, right? And there's also so much structure because your teammate need to have ways of communicating with you, need to know you well enough to make these really flexible yet extremely rapid group decision in a way. On the other hand, we want to maintain unpredictable against our opponents as a team, right? So poker, another great example. If you're fully predictable, you're gonna not do well, right? So I think there is a lot of stochastic element in our decision-making process, while we also trying to learn as much structure about the environment and about others as possible. So we definitely have both, I think, in our decision-making process.
Could you tell us then a bit about the different paradigms then? I'm really curious to know in more detail.
Yeah, I mean, the three of them they're all in the general umbrella of what I described before, but actually in reality, they're quite different. Okay, so let me start with the first paradigm I talked about, which is using Hassett's mouse paradigm in order to understand the neural mechanism of decision on the risk. So economic decision making is a type of value-based decision, and it's usually multivariate. What does that mean? Is that there's multiple dimensions of the options that we should sort of integrate. And then, for example, deciding between different kinds of juice, there might be amount of juice, the flavor of juice, the caloric content of the juice, and so on, right? Think about how from small decision to big decision, most of the decisions are multivariate. And what we decide usually reflect a sort of subjective preference, right? There is no, for example, in perceptual decision making, you have this is the correct choice based on the associated stimuli. So economic decision making has been studied, for a long time in humans, in monkeys, and more recently in rodent paradigms as well. But there's one particular question that we're interested in, which is intuitively you think about economic decision making as an abstract decision making process, right? Let's say which lab to join for your PhD, right? But ultimately, all decisions have to express, almost all the decisions have to express in actions, right? We have a motor plan corresponding to whatever decision we make. And a lot of the times, these abstract value-based decision process and action planning are done simultaneously. Let's say thinking about reaching for an apple or for an orange maybe I make the motor plan the same time I make this value-based decision, right? But in terms of understanding how different brain areas actually represent the abstract value decision process versus action planning process, how they interact with each other, which circuit elements are required for these different computations, we actually thought it was really helpful if we can temporarily separate the two computations so that we can use modern neuroscience tools to dissect the neural substrate underlying one versus the other. The way we're doing that is to play with the stimulus. So we give half-X mouse multiple types of stimulus. And for example, for the first half of the trial, we'll play a sound. And the frequency of the sound will tell the animal on this trial how good the options are. So in this behavior, mice are always choosing between what we call a lottery, which is a probabilistic reward that could be really good or not as good. And then what we call sure bet, which will be 100% of getting a small reward, right? And the sound will tell the mouse on this trial how good is the lottery payout if it's paid out. Let's say 70% of getting 16 microliter of water versus sure bet will be 100% of getting 3 microliter. And on some trial, the sound will tell you on this trial, the lottery is not that good. So when the lottery is not as good, you should definitely choose the sure bet, right? But let's say the lottery is 70% getting nothing, then choosing sure bet is the right thing to do. Let's say what we call expected value, which is reward probability times reward size. Let's say the sure bet and the lottery have the same expected value. Then what do you do, right? Then that really reflects a lot of individual differences. But sometimes the expected value of the lottery is way better than the sure bet. Then as reward maximizing agents, most individuals should choose the lottery, even though there's 30% chance of getting nothing. So we play with the stimulus. Yeah, go ahead.
Quick question. What brain areas are you measuring when you're doing these experiments?
Yeah, that's a good question. The short answer is that I am not attached to brain areas just because in the past, let's say 5 to 10 years, the technology we have can really allow us to survey a lot of brain areas. So we try to take a somewhat unbiased approach at the beginning and then dig in once we have some evidence that one brain area is important. That's not to say that we shouldn't have a hypothesis when we're doing these sort of large scale surveying of brain areas, right? And then I'll get to the point a little bit in terms of which actual brain area turns out to be important. So at the first half of the trial, we gave animal the sound to tell them how good the lottery is. But this stimulus doesn't tell them where the lottery is. And during the second half of the trial, then we give them a different stimulus to tell them on this trial where the lottery is. So then they can combine the location of the lottery and the type of lottery, which is cued at the first half of the stimulus, in order to form a motor plan of, in this case, either leaking towards left or towards right. And the location of the lottery is cued by a whisker stimulation, but it's randomized across trials. So you wouldn't know until you have the cue on this trial where the lottery is. So the important thing is that the design of task, sorry, it's a little bit complicated to talk about without actually graphics to help us. The important thing of the task is that during the first half of the stimulus, motor planning or spatial choice could not be formed because you only know, should I choose lottery or should bet? You don't know, should I look left or right, right? And then that can only be formed in the second half. So this gives us an opportunity to then ask what brain areas are important or required for either the abstract value computation or the motor planning processes separately in time, right? Which was really the power of this paradigm. Yeah, so you asked a great question of which brain area, which is it's a very good question, right? So there are a few primate paradigms that actually do achieve the separation of value from action planning. But most of the other paradigms, both in primates and in rodents, have the simultaneous process of action planning and value. So there's hypothesis about brain areas such as orbitofrontal cortex will be really important for value computation. And then brain areas such as frontal motor cortex or secondary motor cortex might be more important in terms of action space decision. But we all know action signals are dominant and they're really big, and they are, once animals start to plan action, it's almost like the entire brain is doing that computation, especially in the past few years. We see a lot of these types of reports. So action is really important for animals, and a lot of brain area is computed, right? So once you can have simultaneous action planning and value-based decision, I think it's really tricky to identify brain areas that are important for value that also are important for action, because you're going to see dominant action signals, and then you're going to be like, okay, higher motor cortex is action, right? But in fact, there might be value there, but you just can't see it because it's hidden, it's masked. So by separating these, I feel like really give us a great opportunity to try to see what's important in the first epoch, what's important in the second epoch, but more importantly, what is the circuit that facilitate what we call the value to action transformation, right? Mm-hmm. Interestingly, so we started some surveying type of experiments by doing wide field imaging, which is a mesoscale imaging of the dorsal cortex, and optogenetic laser scanning experiments that actually allow us with really high throughput to ask which of the dorsal cortical brain area is causally required, either for the value epoch or for the motor planning epoch. And it seems like still, it wasn't like one. First of all, the caveat here is that we're not looking deep, right? There's a lot of deep regions, such as orbitofrontal cortex, striatum, amygdala, and medial prefrontal cortex, that will all be likely contributing to this behavior, but we're on the cortex for now. We're on the dorsal cortex for now, but we are doing neuropixel recordings that are starting to look at deeper region. But being on top, we're starting with the survey, and it seems like it's not like we only found one brain that is important, but it's also not the case that the whole brain is important for both, right? And surprisingly, and this is all completely unpublished data, this brain area in the secondary motor cortex that has received a lot of attention in the past decade, called anterior lateral motor cortex, ALM, that's being implicated in motor planning a lot. And groups such as Norlee's group and Carlos Sabota's group and so on have really made a lot of advances in terms of understanding the role of ALM in motor planning. So we thought ALM would definitely be important for the second epoch, and it was. But we were really surprised to see that ALM had a causal role in the abstract value decision epoch, when no spatial choice can be formed. And that was really, I think, a novel finding that we didn't expect. In our task, we have the value epoch, and then we have the motor planning epoch, right? And the idea is that there's a brain area that might represent both computation and might facilitate the transformation from one more abstract economic decision to a more specific motor plan. So previously, based on the literature, we had hypothesized that ALM would be important for the motor planning process. But we were surprised to find that ALM was also extremely important, but it was the most critical region for the first epoch, which is the value coding epoch as well. So that was kind of the surprising bit, that was important for both, actually. And that suggests that maybe ALM is a critical node for the value to action transformation. And since then, so the experiments I mentioned before have been more on the mesoscale survey level, where we did either optogenetics or wide field imaging to see, is the information there, and is the information required for behavior? But now we are really digging into the details using cellular resolution, 2-photon CALSA imaging, and neuropixel recording in order to see both on a single neural resolution level, but also on the population of LM neurons in order to see exactly how the representation is like, and maybe combining perturbation methods and recording technique to see, for example, if the value information locally in LM is inactivated or silenced, how does that affect behavior, but also how does that affect the neural population dynamics in LM, where presumably underlie the collapse of the transformation, right? So these are the kind of ongoing experiments that we're doing. But I think to summarize for the first part, really we designed this novel behavior, which is at the beginning quite a challenging behavior to train Helix mice to do, but I have a great postdoc, Ollie Gould, who is really amazing at facilitating the establishment of this novel behavior. But that was the entry point for us to ask these kind of questions. And the survey level techniques allow us to identify this critical node. And now we're digging in and also thinking about how this region communicate with various other brain areas in order to instantiate both the value coding computation and the motor planning process. That's the first bit. Should I move on to the second project?
I also have a question about the first task before you move on to the second.
Oh yeah, please go ahead.
Just from the perspective of somebody who doesn't train animals or do behavioral research, to me, it sounds really complicated. And for a mouse to learn these things, like we were just saying how, it must be confusing for a mouse to be put in this situation, in this paradigm, under these experimental conditions without any instructions and just forced to form these associations. So firstly, how easy is it to train them on this task to initially form these associations? And also, you mentioned seeing variability across animals. So can you in any way kind of correlate the ability for the animals to learn with the outcomes of the task and the neural activity related to during the tasks?
Yeah, both of these are great questions. So thank you for asking them. So for the first one, how difficult it is to train animals to do this. It's difficult. It's challenging. I think at the beginning, we weren't sure if mice can do it at all. I think you really sort of, ping the correct points there, which is it has many aspects of it, and mice need to understand every single aspect and combine them in order to form sort of this knowledge of how the task is, right? So there is this process, what we call shaping, where we teach them very simple things and then element by element, building onto the complexity of the task, right? For example, on day one, they're just learning that licking give them reward because that's kind of how they make choices. And then they learn, we fix the location of the lottery position on every single trial. So essentially for the animal, they're just ignoring that information because that doesn't give them any task relevant, reward relevant information, right? Instead, what we first teach them is the sound that corresponds to the value of the lottery. So if they can learn that, then we start to gradually change the position of the lottery at the beginning a few days at a time, and then every other day, and then within session, and then finally fully interleaved. So you can see how we first teach them the sound, then we teach them the meaning of the whisker stimulation, which is the lottery position cue. Then we ask them to combine everything. Still, it's a pretty complicated task, and does take a few months for animal to learn. And I think, the devil's in the detail when it comes to animal training, right? Like we really should know. I've heard this before, like if animals are not learning, it's not because they're not smart, it's because we didn't teach them the correct way, right? Because indeed, like you said, we can't tell them what to do. So they learn everything from reward feedback, right? So we have to design things so that they meet each goal and then they can combine everything for the final task. And the individual differences is extremely interesting, and it usually reflects not only in terms of how fast they can learn the task, but also in terms of things they learn after they learn the task. So they do the task, for example, for a few hundred trials within the period of, let's say, 1 hour, right? But not every single trial is the same. At the beginning of the session, so these are water restricted animals that we use water for reward. At the beginning, they might be extremely motivated, right? And then their preference might be slightly different compared to in the middle of the session when they're still motivated, but they're not so desperate, versus towards the end when they're satiated, they might still look left and right, trying to see what they get, but maybe they're not trying to be real maximizing on every single trial. So, and there's a lot of individual variance there, right? Because you can imagine it's a matter of attention modulation, arousal, and motivational state, and how well we do our task is a function of motivational state. Even in humans, right? Some individuals only perform well when they're motivated, and but don't perform well when they're not motivated. But other individuals might work hard despite whatever the fluctuations in the motivation. Mice can be the same. And one way that we're thinking about this and dealing with this in a way is to use unsupervised method in order to identify sort of behavioral state when animal is engaged in the task and is actually doing the task according to the stimulus we give them. And then we can identify the trials that belong to these engaged states and analyze that as a more sort of homogeneous behavioral state / strategy that we can identify across individuals. But then the rest of the trials, what we call disengaged states, either they're just biased towards one side, or they're always choosing sherbet, or they're always choosing lottery. These are quite different across individuals. And so far, we just lump them together as disengaged states. We can compare the two states when it comes to neuroactivity, for example. But digging into the individual differences, I mean, that's the end goal, I would say, to really understand individual differences in decision making and actually map the neural mechanism to that. That would be the ultimate goal. As you can imagine, it's hard to do, but I think that's always a really interesting question. And especially when we're studying sort of decision preferences, we see that a lot, right? I don't know if that answers the questions.
Yeah, definitely. I think as somebody who finds it a lot easier to think about things in a reductionist sort of way, in a reduced environment, I really admire the ability for you to carry out these experiments and keeping in mind all the variables that there are, all of the inter-individual differences, everything, and just using that as a benefit to say, look, we have the data nonetheless. You can manipulate it in this way. You can use an unsupervised approach to shape it differently and answer different questions, but like this is the paradigm and this is what we're testing, and we can be clear about these certain things and maybe less clear about other things. So yeah, really great
I
answer.
think, there's always constraints, I think, with each approach. And then the second project is kind of in between the very controlled and sort of let it go sort of approach, where we have still have fixed mice playing with a computer opponent that the computer opponent we control, right? But it's an interactive process. So basically, still have fixed mice looking left and right. The computer opponent tracks the choice and reward history of the mouse, so far what they have done and their reward history in the past session, let's say. And then they're trying to predict, the computer opponent is trying to predict what the mouse is going to do next based on the past patterns. And if the computer predicts correctly what the mouse is going to do next, the mouse is not going to get a reward. If the computer failed to predict what the mouse is going to do next, the mouse gets reward. So as you can imagine, with this kind of competitive pressure, mice slowly learn to become stochastic and sometimes really close to fully random playing against a computer opponent. And, there are people who study versions of these type of questions. Dale Lee have been studying these kind of questions in monkeys for a long time. We learned a lot from these studies. Arkhipova, her group has been thinking about this in freely moving rats, and more recently, Alex Kwong's lab have been thinking about this in high-fixed mice. But there's some challenges when it comes to using neuroscience tools to understand strategies, because strategies, for us to estimate strategy, we need many trials, right? Because based on these trials, we're like, okay, that's the strategy you're using. But the modern neuroscience tools is really rewarded for resolution, temporal resolution, spatial resolution, and so on. So we wanted to have a single trial estimate of what animal strategy is. And if we can do that, then when animal learn to become from very low noise, low entropy, non-stochastic type of things, which is what they usually do on day one, they just lick one side, and then the computer fully predicts it, they have no reward whatsoever, and after a while, they switch to the other side. And beginning, that was surprising to the computer opponent. They get a bunch of reward, but eventually that was also fully predictable, and they get no reward, right? That's day one. And over the course of 2 to 3 weeks, it's a really mysterious thing. Like, how do they learn just by licking and getting water or not reward water to eventually become almost fully stochastic, where the computer can almost never predict what the mouse is doing, right? The entropy of their choice reward pattern is extremely high. And it really doesn't seem like they're doing this by not paying attention or zoning out, because as soon as we turn off the computer opponent, they go back to looking to one side, because they were like, okay, now I don't have to be high entropy in my behavior, so they revert back. So again, we're trying to use unsupervised method, modeling method, in order to characterize or describe animal strategies and be able to sort of attribute a strategy to every single trial. And then obviously with different levels of confidence. And now we have this tool, we can track neural activity during the entire learning process and try to see what are some of the neural mechanisms that will give us a clue about how animals learn to do such a thing, which is, by the way, difficult for humans to do as well. If you ask a human to produce a random number or, picking left and right randomly, what we have is what we call a wrong length, a problem where most people think it's impossible to have, let's say, 8 left in a row according to a random process. But of course, it is possible. So what will end up happening is that the distribution is shifted, so it's not fully random. But if you ask humans to play against the computer opponent, you can see also their stochasticity kind of increasing. Again, we have the super interesting behavior paradigm. And right now we're digging into the neuroscience. My first PhD student, Joanna Allure, really spearheaded this project. And there's some hints now. We have some preliminary results that seem to suggest really a dissociation of reward and current choice. Instead of following which side give you the reward, which is what reinforcement learning is, right? Usually for learning, we should follow which option is good. And if this option is no longer good, we switch. But that's not the right strategy here. So what animal learns to do mysteriously is sort of a dissociation between what I earned in the last trial and what I should do on the current trial. Trials become much more independent and reward information is decreased so that you're not trying to follow a teacher anymore. You're trying to, because as soon as, at the beginning, we thought maybe the animals, when they learn, they're testing different hypotheses, but the more confident they are with any hypothesis, the more they're getting a punishment for that, because then they're going to have structure in their behavior and that will be punished until they become fully stochastic. So this, again, is a challenging project because how do we describe the behavior? There's no sensory stimulus. It's really about their own choice and reward history. And how do we understand random behavior, right? I mean, the approach we took is to compare random behavior with very structured behavior, and the contrast between the two is kind of telling us something instead of saying the absolute random, what does that mean? That's a very, very hard question to pursue. Yeah, so our preliminary data is again using wide-field imaging throughout the learning process, but we're excited to dig into neuromodulation as well as sort of single neural resolution recording or imaging throughout the entire learning process. Super excited about this project as well. It's just like good dinner conversation, all of the projects in the lab,
Yeah. It's so awesome.
Yeah, that is like one of the most fascinating things I've ever heard. Like,
Thank you.
the like really probing stochasticity is just something I've never really thought about doing. And I think it's so interesting because it brought to mind like just the idea that most of the things that we do and experience through our lives were mainly reinforced. Like we mainly have to learn rules and learn how to behave and keep in line with that, especially being very social beings.
And so much so that it's challenging. It's almost impossible to be intentionally random.
Yeah. I mean, I used to think so, but now I'm kind of taking an alternative approach to thinking about it, which is maybe stochasticity is extremely fundamental. We just maybe don't think about it that way. I have this friend, Dimitri Arnov, who work on birds, and we kind of make jokes about this when I talk about this behavior to him, and then he's like, oh, that's creativity, right? Like any sort of deviation from structure, any sort of deviation from patterns that we already know are new things, right? Are unpredictable, not only as an advantage when we're competing with others so that we can maintain predictable, but also from our own perspective, right? Like I had a master's student who was a jazz player, and he's like, I love this kind of thinking about how to study this because this is what I do when I'm playing jazz as well. I think it is quite fundamental. I think it's true that not a lot of neuroscience is focused on this. So definitely that's the case, I think. That's why at the beginning, even realizing how challenging it is to study this, because we don't have really great theoretical framework to understand non-reward guided behavior or anti-reward guided behavior, which is something that we're trying to do ourselves. But I never wanted to give up on this project because I think once we have a grip on it, it's like uncharted territory that for us, of course, we should understand this fascinating aspect of our everyday behavior.
Yeah, it's
Yeah.
so cool.
Wow, I love that point about creativity.
Yeah.
And also because I was thinking when, again, I think this whole episode has just been me trying to pretend I'm a mouse under one of these paradigms.
Sure, yeah.
I was thinking if you're put in this paradigm where no matter what you do, it's sort of working against you in a way, and you have to it's hard to tell what really will help you get the reward. Will that make you more motivated to kind of figure out what is the thing that will get you the reward, or will you just lose
Disengage,
interest?
yeah.
Yeah, that's a good question. So from both behavior and neural data we have so far, we definitely know they're not disengaged. So one idea of knowing that they're not disengaged is when we make environmental changes, such as turning off the computer opponent, they instantly realize that and then adjust their strategy back to a strategy of licking one side, which presumably is lower energy, sort of less demanding strategy, because when they don't have to be stochastic, they're not, right? The other thing is that we see a lot of sort of preparedness sort of increasing when they're doing the stochastic strategy. It's too much to go into details about this, but at least showing that it's not like they're zoning out or kind of giving up. Instead, exactly where is the source of randomness is the aspect of this project that we haven't dig into, by the way. That is how is random behavior generated? Where is the random number generator in the brain? That's the question that we haven't started to dig into, but it does seem like our data seem to suggest a dissociation between reward and action is also a prerequisite for animals to be stochastic. Yeah, so that's one of the questions we always get, right? Like, is random just giving up? And it really doesn't seem to be the case. If anything, it seemed to be a higher energy kind of thing. And I think we were mentioning this before, when this is a good thing to have in our behavior, one of the scenario is in dynamic multi-agent competitive environment where you have to be stochastic. Again, a higher energy type of thing where there's a good incentive to be stochastic, right? So yeah, but these are great questions that we're thinking about for sure.
You mentioned that you used unsupervised tools to categorize the behavior. Could you tell us a little bit about what the strategy is, if you do know what it is? I'm assuming it's not just, randomly left, right, because the computer would pick up on that. So what is the strategy that the mice use to be random?
That's a great question. So you're right, because the way that our computer opponent work is that it will look back, one trial back, two trial back, three trial back, four trial back, both for choice and for choice reward interaction. So if the animal is doing left, right, left, right, left, right, left, right, that will definitely be picked up. And that will be, animal will get no reward for doing that. So if you count like 1 to 4 trial back, reward and choice and choice reward interaction, that's a really big map of potential patterns of behavior that animals can do and be picked up. So what does the animal do, right? So how do we identify when they're stochastic? So I guess there's two aspects to this. One is the more fundamental aspect of there are certain stereotype strategy that animals tend to do. For example, bias, which is just biasing to one side. The other one that's very common is what we call perseverance, always choosing what you choose on the last trial. And the other thing that's quite useful in many other tasks, but not so good in our task, is win-stay-lose shift, right? So you repeat action that was rewarded on last trial, and then you switch from the action that was unrewarded on last trial, which is a pretty good strategy for learning. But since the computer can pick up 1 to 4 trial back choice reward interaction, win-stay-lose shift will be picked up as well. So animal cannot do that. The way that we're identifying their strategy, I keep mentioning unsupervised method, is this newly established and popularized strategy called GLM-HMM. So the idea is that animals have distinct behavioral states, and they switch between these states. Each state might correspond to tens to hundreds of trials, and each state corresponds to distinct combination of different GLM inputs. So when people use GLM, initially was used to characterize perceptual decision making. One of the GLM input could be the stimulus, which is maybe the most important task relevant dimension. And how they identify engaged state will be a state that have very high weight on the stimulus, but very low weight on any other sort of biases, history-based bias or left-right bias and so on. So that's what they call engaged state. And the other disengaged state will be, for example, a bias to left state that actually has a lower weight for stimulus. They're ignoring stimulus, but going to sort of left versus right and so on. In our case, we have no stimulus. So I have to think about what should be the GLM inputs in order to identify these different history-based strategy. So we have a very simple 3-state, 3 GLM input GLM measurement model where we can identify bias strategy, perseverance, Wednesday rule shift, and we simulated fully random strategy, and that will come out as 0 weights on any of these inputs essentially. And then we first generate synthetic data in order to validate the methods that it can pick up. Indeed, we generate ground truth synthetic data, and then we ask the method to identify it when it's mixed together, and our method can do it, which was a good thing. Synthetic data is very useful before we try to use this for mouse data. And when we applied this to mouse data, we were actually quite surprised because we were imagining animals to have different complicated history-based strategy and then maybe also a stochastic strategy. But instead, the most dominant 3 strategy coming out of the animal is bias left, bias right, and fully stochastic. And we have to say, the fully stochastic is with air quotes, right? Because that just means we couldn't identify any of the things that our GLM inputs can identify. And then we train the computer opponent on the trials that we identify using this to see if the computer opponent, which is much more powerful than the GLM inputs that we give, can identify any patterns that we didn't identify. And the computer opponent also was at chance. So it really seems like the stochastic trials that we identify using this method are, at least against the current computer opponent, unpredictable. So that was kind of like, in a nutshell, how we use unsupervised method to identify the strategy. And I should say that it's not a perfect thing, right? There's many things that we can improve on. For example, how do we decide how many states is the right number of states? The fitting process has the devil's really in the details. And also, when fitting to learning data, we shouldn't assume that states are constant throughout learning or the transition matrix between the states will be constant. So there's some judgment call to make there, either to simplify the method, to make it more interpretable, or to expand the method so that it elegantly captures everything, but then potentially become less interpretable. So these are definitely the balance that we play with. But I think we're pretty lucky because, GLM measurement has been popularized over the past let's say 3 years. And before then, I think we might struggle to identify this single trial level resolution of what kind of strategy anyone is using.
So cool. Can we talk about the third paradigm?
Yes, the third and the final. I'm so sorry that I'm taking so much time from you guys.
No, not at all.
Okay, so the third paradigm, which is the paradigm that for the longest time I wanted to study but didn't know how to. How do we try to understand value-based decision-making in an interactive, real to animal scenario, right? So we call it social decision-making, but I think really we're trying to study value-based decision in a multi-agent context. So we have this paradigm that we call the octagon. It's a very dynamic foraging environment. Octagon, as you might guess, there's 8 different parts of the arena where animal can go and forage for reward. And on each trial, 2 out of the 8 what we call patches are active. But the position of which of the 2 of the 8 patches are active change across trials. So really, you have to pay attention to the environment to know on this trial, which of the 2 patches actually has reward, right? And then we first train individual mice to associate a certain visual stimuli with high reward and another visual stimulus with a low reward. So you can imagine on this trial, 2 of the 8 patches have visual stimuli, which means you have to go there for reward. One of them, let's say, has grading as pattern. The other one has light patch as pattern. And animal learn via reinforcement learning over, let's say, weeks that grading has high reward than light. No matter where it is, if I see grading, I should go there because that's the high reward patch. So here they really have to generalize this value, stimulus value association beyond position, right? Because position actually doesn't matter here. It's really the visual stimulus that you have to pay attention to. Even that behavior on its own, I've never actually previously studied these kind of navigational based behavior in such a large arena. Even that on its own is so rich. I was kind of like a kid in the candy shop in a way, thinking about what we used to complain about, right? The control behavior that's hard to control. They're not all natural behavioral variants in terms of animals' position relative to one or the other patch, how that influences behavior. Do you go to the closer one, or do you perfectly respect value instead? Animals' head direction, whatever they see first, also especially early in learning, can bias them towards one versus the other. Animals' trajectory as they travel towards the goal, in a way, is like an embodied readout of their internal decision process, which is amazing, right? Like, I'm a big sort of fan of thinking about embodied cognition, and I see that I'm like, not only am I not worried that we need to control that, but I'm actually happy that this provides a window to animals' internal decision process. Yeah, so that is already a very rich behavior, and I was really happy with that. And animal indeed can, over weeks of time of training, be able to develop a preference for the high reward patch, no matter where the patch is, no matter where they are in the environment. So that's the first step. And I've been thinking about how to kind of study multi-animal decision making for a while, and then I take a lot of inspiration from, sort of classic game theory tasks, maybe in humans, and also my one of my academic heroes, Dave Williams, and when Karen Harouche was in his lab together, they developed a prison's dilemma in monkeys. I was like, oh, I wish we could do something like that in rodents. So we had a lot of big dreams, and that's why we designed our paradigm this way. It's logical because animals can freely move around, interact with each other and with the environment, but also controlled because the stimuli is controlled and the reward delivery is controlled and so on. But in the end, I sort of decided against a very, one half-X animal playing with another half-X animal with a very dry sort of reward payoff matrix, kind of classic game theory sort of thing. We decided to go for a simpler game, but allow animals natural behavioral variance to sort of give richness to the game. So the rule of the game is, as I mentioned before, once animals learn about the rule of this dynamic forward environment, we put 2 of them together. So they both know how the environment is, right? And like I said before, when they're by themselves, there's the high, there's the low, and they have to look around on each trial to first know where the patches are, and then go towards the high patch, which is their learned preference. But then now the rule is the faster mouse is going to get the reward of their choice. Could be high, could be low, whatever their choice is. And the slower animal will get nothing, right? And because they couldn't predict where the patch is going to show up, it's random on each trial. They can be anywhere in the environment relative to each other, relative to the goals. And suddenly 2 patches show up. One of them could be really close to one of them and far away from the other. And they themselves could be close by to each other or far apart. Each will provide different sort of competitive nature, fairness, uncertainty, which make each trial unique. And which also means high, low, and no reward now became the potential outcomes. And that really depends on the ongoing behavior of both self and other, right, of their opponent. So what I say expected value, which is reward size and probability, now the expected value changes on a trial by trial basis, depending on the ongoing behavior of self and opponent. So that's really, I feel like, the football kind of decision making, right? It's flexible, it's rapid, it's multi-agent, and it's in this really fast-changing environment that you know some rules about, but you have to apply this knowledge in a very flexible yet very rapid way. And importantly, not only do you have to think about yourself and the environment, which is most of the decision-making studies that we've been doing, right, including myself, we have to think about the relationship between your opponent and the environment and you and the opponent, right? So the first thing we set out to do is just to simply ask, both by analyzing the behavior data, but also modeling the behavior data and using artificial agents to simulate multi-agent competition, to ask the question of, do mice integrate information both about self and their opponent in order to flexibly guide their choice on a trial by trial basis to maximize reward? And our preliminary data suggests that all of the things I just said are the case, which was every time I do analysis and it works out, I'm like really surprised. Do animals pay attention to the other animal's position on a trial by trial basis to decide what they should do on this trial? I wouldn't have put a bet on that to be like definitely the case, but that seems to be the case. And again, the interesting thing is that there's individual variance, right? So not every single animal does that. So there are animals that pay attention to others more than other animals. And now we're digging to the really interesting thing to see what aspect of the opponent do they pay attention to. Position is one of them. Turns out speed is another one, which kind of makes sense, right? Because it's really a competition of time, right? It's distance, but if you're fast, then that means different compared to if you're slow. If they're playing with, let's go for the extreme, an opponent that's infinitely slow, which doesn't move at all, you should just behave as if you're by yourself. Versus if you're playing with a fast opponent that really can change the outcome of your choice on every single session, on every single trial, then you should behave very differently when you're competing with this opponent compared to by yourself. So these are the kind of questions that we're asking now. And it's just been extremely fascinating digging to how animals play with each other with this extremely simple rule, which is just competition. Winner gets the reward and loser gets nothing. But how many sort of source of information they integrate in their decision-making process. And then we're also getting to the neuroscience of it, which obviously has some challenges because multi-agent, freely moving, navigational, but we're all getting there. I think, the next 1 or 2 years, it's going to be extremely fruitful in terms of getting the data, but also understanding the data of, starting from brain areas such as prefrontal cortex, but also thinking about brain areas potentially as the superior colliculus, hippocampus, and so on for different questions in this paradigm. And before I open for questions, we're designing almost the same game as a video game for human subjects in order to first starting with first person video game, but also maybe down the road thinking about VR to see, again, without giving them instruction, letting them learn the way that mice are learning by feedback and learning about the relationship between their action and reward in order to, first of all, learn the task, but also then play with another human being or potentially a computer opponent that we can design with different strategies and see how do humans behave in this environment? What are the individual differences there? And if we do have a very interesting behavior data there, we might go into dig into neuroscience as well, collaborations. And, I really love this sort of cross-species approach of thinking about neuroscience. My lab is almost completely mice, but I'm very excited with this small, but potentially going to become larger dimension of human experiments as well.
Super cool.
That's great because I was actually going to ask I wanted to kind of bring it back to the human side, given that you started with your interest in human psychology. And so, that's really great to hear that you are thinking of translating that, because I think the benefit of studying such complex behavior in mice, of course, you want to do that to get to the neural mechanisms, but the benefit is also that you can then translate it back to humans. So, really great.
Definitely. And I think in the past few years, there's a sort of, because humans can extract information from words and from language, right? So a lot of the human tasks are with words or with stimuli that's sequential on the screen. But over the past decade, we see a lot of new studies that think about embodiment when it comes to decision making. And, we have studies where people are actually physically walking around, some maze. And the video game is just kind of an intermediate version of that. And I really like the idea of embodied decision making in humans, because we don't do that always, but we a lot of time do that, right? So
Yeah.
I think, again, going back to the football idea, it's embodied, and that potentially involves a slightly different network of brain regions, or potentially even different underlying computation when you have to integrate this information, but then transform into action both flexibly and rapidly.
I had a technical question. When you deliver the reward and some is large, some is small, you mentioned like different light patterns, are those always the same in relation to the reward volume, or are they also random?
Yeah, so the association between the visual stimulus and the reward mapping is fixed within an individual. But of course, across animal to control for sensory biases, we will kind of counterbalance the mapping. And also my postdoc Ivana and I are talking about, and she's tried moving stimuli. So the way that we set up our paradigm, I didn't have time to talk about it, is that we did it so this is in collaboration with NeuroGears. And we did it to be very flexible, so that instead of, let's say, LED or something, what we have is this sort of projector that projects visual stimuli onto the floor of the arena. Because of the projector, then you can project anything. Like you can play a movie in colors if you want. So far, we started with very simple stimuli. Grading versus light is what we started with at the beginning. But Ivana has tried moving stimuli, so moving checkers versus moving grading. So yeah, I mean, at the end of the day, it's really about them learning value, and the sensory aspect should be balanced and distinguishable. So these are the things that we think about when we design this. But this is a good question, by the way, because when I first started this project, every single thing we have to kind of establish, right? Like what kind of visual stimuli they can learn on the floor when they're moving around, where they might not be always looking at it. So all of these, every single decision that we have made in designing the task is part intuition and part experience, and part experimenting, which is kind of the fun bit. But yeah, so it's been really exciting so far, for sure.
It's been amazing listening to you and all of the really cool paradigms that you have set up.
Thank you so much.
I'm so excited to read more and hear more about your research in the coming years. Thanks for joining us.
Thank you.
Yeah,
Thank
I'm
you so
really looking
much.
forward to seeing everything that comes out of your lab in the future. And thank you so much for your curiosity into this
Thank you.
topic.
Thank you so much. It's been really fun chatting with you guys.
Thanks for listening to today's episode. If you enjoyed today's episode, please feel free to share it with your friends. Our social media @ are neuroverse_pod, and check out our website for resources, neuroversepod.com. Today's episode was made possible thanks to the support of the Sainsbury Wellcome Centre Public Engagement Fund. We would like to thank Sainsbury Wellcome Centre for the generous grant supporting science communication initiatives like these.